All right, welcome back. So we're going to continue on with chapter 2, and now we're going to move on to the macromolecules, and I'm going to break it up into 2 parts. So today on this one, we're going to talk about your carbs and fats, and then the next lecture we'll talk about the proteins and the nucleic acids. So first off, organic chemistry. Basically, it's the study of carbon. Carbon is like the molecular tinker toy for biology because carbon has like this perfect number of electrons in its outer shell to where it can interact with 4 different atoms at once. And it loves to form these covalent bonds, which are really strong. So you will see carbon show up in all the different macromolecules that we're going to talk about over these next 2 lectures. Organic chemistry, if you know someone who's taking it, just give them a hug because it is a really, really hard class. I'm just telling you from experience. So before we get into talking about the nitty-gritty, let's introduce a few terms. So the first one's going to be monomers and polymers. Now, a monomer is a molecule that may bind to other molecules. It's an individual unit. So example right here, this right of our little molecule right here, this is a monomer, the individual unit on its own. Now, when we start to build a chain of them, so similar things, so it's the same unit over and over and over again, that's a polymer, and they're linked by these covalent bonds. So monomer, mono is the one, polymer is multiple units of the monomer. So it's really simple when you think about it this way. So poly is multiple, mono is one. Now, how we make these reactions are going to be either called a condensation reaction or a hydrolysis, and it's going to involve water. So if we're going to build a polymer, we're going to have our 2 monomers right here. And what we're going to do is we're going to remove 1 molecule of water. So water is going to leave, and now we're going to link these 2 via a covalent bond. Now this is called like a dehydration synthesis or a condensation reaction. So think about like you have a cup of ice water. Think about the water that forms on the outside of your cup, the condensation. Water is leaving, it's on the outside of it. It didn't go through the glass, it just kind of condensated from the atmosphere. Water that was there. Now, if you want to go and break the reaction down, it's called hydrolysis. So here's our polymer right here. So if we had to remove water to make the bond, in order to break it, let's just add the water back. So hydrolysis is adding it back in, and now we have back to their 2 monomers. Now you're going to see there's 4 key groups. This lecture is going to focus on the first 2, and the next one focuses on the next 2. So the first one are carbs, and these are your sugars. This is probably one of your favorite ones. If you look at this, there's probably a lot of good things you love to eat right there. Now, it can be as simple as a single glucose molecule or the complex carbs, which contain thousands of monomers together. They have so many different functions too. It's not just a simple source of energy. Now, it's in a lot of the foods you eat. You've probably heard of good carbs versus bad carbs. Now, good carbs, a dietitian would tell you to eat these. It takes more energy to break them down. So it's not like a quick source of energy. Versus a bad carb, which are going to be these refined ones. They're going to be highly processed. They break down really fast. And then your body uses it up, and then you're hungry again. So it's like Chinese food. Think about Chinese food. It's so satisfying, but then next thing you know, you're hungry in about 2 hours. Good source of bad carbs. So if you ever gone into nutrition, your good carbs versus bad carbs. Now, what do they do? Your sugars or your carbs in general, they store energy. So we store it as glycogen, plants store it as starch. So we never know when we're going to get our next meal. So sometimes we like to hoard things. So plants, they will make starch molecules and they'll store it there. For us, if we don't use the glucose molecules right away, we'll convert it to glycogen and we'll keep it until we need the reserves. It can also provide structural support. So for some plants, or all plants, it makes up cellulose. Some animals, it makes up their exoskeleton. And it's important for bacteria. It's part of their membranes. So you probably heard of the keto diet. This has really been a big trend the last couple of years. And what the keto diet is eliminating carbs. as a source of energy for your body. So let's talk a little bit about some basic carb facts. So here's the basic formula. You have the CH2O, and I have the parentheses around it. Basically, that's saying for every carbon, there's one water molecule associated with it. The little n is for how many of these individual subunits you have. So you'll see the term glucose show up. It's going to be C6H12O6. Basically, glucose is 6 of these little molecules right here. Now, if it's a single sugar, it's going to be called a monosaccharide. So saccharide is another term for sugar. Mono, one sugar. It's only going to be a one ring. So here's some examples. As I just mentioned, glucose. Here's fructose, galactose. If you hear the O's, it's probably a good indication it's a sugar. Now, if we want to make it into a polymer, we can do as simple as a disaccharide, so 2 sugars. Sucrose, table sugar, is a good example. So we join 2 monomers right here together. Now we're at a disaccharide. You can have oligosaccharides and all the way up to polysaccharides. It just depends on the number of monomers you have. Something important, structure will determine function. I'll say that a lot because how the molecule is organized, or especially the polymer, will determine what it's going to do. So let's talk a little bit about the monosaccharides. They're simple sugars. They're going to be key for a lot of things that we're going to talk about. So they're going to provide chemical energy for your cells. So glucose is going to be like the input that we're going to use for cellular respiration, and we're going to use that to make ATP, recharge our ATP batteries. It also serves as part of this molecule. So the molecule that we're going to be recharging, a simple monosaccharide, makes a part of it. So this is an ATP molecule that I'm circling on. This is a ribose with a nitrogenous base and 3 phosphates. It also is essential for, right here, here's a ribose molecule right here. So it's a part of a building block for your nucleotide. So nucleotide is just a sugar with a nitrogenous base and a single phosphate. And with each of these different nucleotides that we'll talk about in more detail, the sugars are a key component. Now, polysaccharides, remember, it's just a long chain of monomers joined together, have various functions. They can be a source of energy storage, so starch or glycogen, which I just previously talked about. It can provide structural support. It could be used for cell recognition, how cells communicate with each other. How we form them is by removing those water molecules, so the dehydration synthesis. Now, if you look at this diagram right here, as I'm kind of circling over it, if you look at the bottom row right here, you can see there's like different organization methods used for the polysaccharides. Now, if it's really straight and rigid and highly organized, it's a good example. It's going to be very, very structurally supported. Cellulose is organized like this. Versus glycogen. Glycogen, we want to package it into your cells and we want it to be very compact. So by forming these multi-chains, we make it easier to access it quicker, but it also doesn't provide as much structural support. So depending on what it's doing, so cellulose is going to be serving as protection in their cell wall for the plants, versus glycogen is just an energy source for us. If you look at starch for plants, our amylopectin, you can see it's more not as structurally rigid as a cellulose, so it's easier to access. So just showing you a little bit about cellulose. Remember, it's those long chains, boom, 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 boom. And if we kind of know about, how the organization is super important, so let's look at the cell wall a little bit more. So we have a plant. Here's the individual plant cell. So if we go into the cell wall, you're going to see these microfibrils. And if we dissect the microfibril individually, if you go into the cell part of it, you're going to see these crystalline structures of cellulose. So these long chains that are highly organized lead to rigidity, and it gives plants their ability to grow tall and provide a lot of structure and protection in that cell wall too. very similar to cellulose is chitin. It's going to have the same organization. It's going to have that rigid structure. Now this is going to be in the cell walls of some fungi, algae, arthropods, cephalopods. It is very abundant in the world. Now it does make up the exoskeleton of some organisms here. So, you know, like when you crunch a bug, you hear that squishy sound. Well, the exoskeleton is given its protection, and that protection with a bunch of sugar molecules, which is called chitin. And when you're crunching it, you're kind of squishing some of those bonds right there. As I mentioned also, they can also form glycoproteins. So we're going to talk about this membrane layer here in a little bit, but this is your phospholipid membrane. And these long chains right here can form what we call glycoproteins. So what happens is these long glycogen chains attach to proteins that are embedded in your membrane. And they can act like several different things. They can act like a little billboard and say, hey, this is type of cell, kind of give you a status on how the cell is doing, the physiological state. Sometimes it serves as a surface receptor. It has to look for that glycoprotein receptor, or it serves as an attachment point. So this is another thing where a sugar molecule can have multiple uses in the cell: a source of energy, source of structural support. This is a source of communication. Now our next big group is lipids. Now the lipids are going to be a little bit different. So if you remember, I talked about the monomer and the polymer. So remember, the monomer for your carbs is the monosaccharide, and then we have the polysaccharides. Lipids are a little bit different. They're very diverse, and you're going to see a lot of diversity in there. You're going to see the fats, oils, and steroids fall in this category. Now, one key thing with lipids is they're not polymers, and their structure is not composed of a chain of monomers. So you're not going to see like an individual monomer repeated over and over again. But you will see these things called hydrocarbons, which is going to be this long chain of carbons that are going to be saturated with that. And you'll see them in different combinations. So it can be simple chain, linear chain. It could be kinked. You can have multiple chains like a triglyceride or a phospholipid, or that chain can be folded into rings. Now, one key thing about lipids, these are your fats and oils. They're non-polar and hydrophobic, so water-filling. Now, there's a lot of different structures just from what you see from this figure right here, and they have lots of functions. So we'll hit on a few of them. Probably the one that's going to be the most important is the phospholipid. Now, a phospholipid is going to have a unique structure. It's going to what we call this amphipathic molecule. Now, it is going to be composed of a phosphate head group, phosphate and glyceride, and these two hydrocarbon fatty acid tails. Now, what's unique about this molecule, it's going to have a part that loves water, so hydrophilic, so philic, loving, and also a hydrophobic, so the tails hate water, but the head group's going to love it. Now, when you take a bunch of these phospholipids together and like put them in a jar and shake them, what they're going to do is they're spontaneously going to arrange into what's called a bilayer. So all the parts that hate water are going to face each other and form like a no water zone. So no water out in this area. All the heads that love water will face the outside part. This is going to form what we call a phospholipid. It's a component bilayer because you got two layers right here. Now it can also form what we call myceles. Let me get my mouse positioned better, which is just a circle of phospholipids where the tails face inward, heads face outwards. You can use this to move things in and out of the cells too. Now these phospholipids do make up cell membranes, which is super important because everything's got to have a cell membrane if you're a cell. Now your body is mostly water, so you're going to have an aqueous environment outside and inside of your cell. So the hydrophilic heads are going to interact with the aqueous environment inside and outside of your cell. If you want to think of the phospholipid bilayer, think of it as like a barrier, like a city limit for your cell. It's going to be very important because it's going to control what goes in and out of your cell. Now, We'll talk about transportation in and out in the next unit, but it's going to regulate what goes in and out easily. And sometimes, depending on the type of molecule, you might need a little bit of help. If you're a nonpolar molecule, like oxygen, carbon dioxide, it can go through the membrane easily because remember, they have to go through this no water zone. But if you're a hydrophilic molecule, one that loves water, this phospholipid bilayer is going to say no because you can't go through the no water zone. So this is where you're going to have to probably get a little bit of help. And that help comes in the term of proteins, which we'll talk about in the next lecture. So proteins are going to be embedded in this phospholipid bilayer, but it serves like a transporter, like a little channel to help move things in and out of the cell. So if you're a hydrophilic molecule and you can't go this way, you're going to use these transport channels that are made of proteins to help you move through it. Another component of your membrane is cholesterol. So you've probably heard about cholesterol. It's one of those things I tell you to regulate in your diet. Having some of it is good because it is part of your membrane, helps adapt to changes in temperature and then make sure that they're very fluid. All right, your steroids. So remember, we're talking about these hydrocarbon chains, which were very similar for the fats. And steroids, they kind of fuse these into rings. So when you think about your steroids, think of your sex hormones and cholesterol. They both fall into this category, and they have various functions. So I just mentioned cholesterol. It's very important for your membrane, but too much can be a bad one too. Steroids, they're very important for regulating a lot of activity in the body, but abuse of them also does horrible things to the body also. Another member of the lipid family was the triglycerides. So basically a triglyceride is a glycerol, 3 fatty acids. Boom, 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 boom. Now it's a type of lipid used for long-term energy storage in the body. So when we need it, this is a good source for energy. So we have our glycogen, which was a storage of carbs. We can have to also store energy in the form of these triglycerides. Now, it's going to be stored in your fat cells, and when your body's going to need energy, it's going to trigger the triglycerides to be released between meals. Now, it's another thing that we need to make sure we regulate how much your triglyceride levels. So when you get older, to get blood work, they're going to measure all these different things, and they've shown that too high of levels also does have deleterious effects on your body. And then these last 2 slides kind of talk about these fatty acid tails and some characteristics of them. So you probably heard of saturated versus unsaturated fats and then trans fats. And basically it all depends on the fatty acid tail. So if the fatty acid tail, boom, this hydrocarbon tail, has all single bonds and it's absolutely straight, it's completely saturated, it's going to be called a saturated fatty acid. So all the bonds are single, and every bond that has an open spot is occupied by hydrogen. Now, an unsaturated fatty acid is going to have at least one or more double bonds. Now, when a double bond happens between the carbons, what it does is it causes a kink. All right, so it looks like a little bend right here. So when that little bend happens right here, it kind of regulates how the molecules can interact. So a saturated fatty acid, since the chains are flat, they can fold on top of each other, stack on top of each other, and then you can make these solid at room temperature. Versus your fats that are liquid at room temperature, it's thanks to this kink in the tails that prevents them from lining up and stacking up all nice and pretty, so it's going to stay liquid at room temperature. Now, man, we love the things. We've kind of figured out how to make that unsaturated fat look more like a saturated one. And that's what we're going to call a trans fat. So remember, there's normally a kink thanks to that double bond. But what a trans fat does is we went through and kind of twisted this molecule a little bit to make it mimic more like a saturated one. And what that does is it allows it now to remain solid at room temperature. So margarine, peanut butter, shortening. they're good examples of that. So yes, it does have that double bond that would cause kink, but since we kind of modified it a little bit, now it mimics more of a saturated one. And once again, this is another thing they tell you to kind of regulate. So I'm going to stop this one right here, and we'll pick up with proteins and nucleic acids on the next one.